My lab is currently structured so that we have a technician and we have 4 other individuals. There are 2 postdoctoral fellows, and we have 1 graduate student, and there's 1 undergrad who's doing a summer research project, and all of whom I've been incredibly impressed with, and it's great to work with them. One of the basic aspects of what we need to do is to grow viruses, to see how well they grow or how well we can keep them from growing. And that involves keeping stocks of the virus, which we'll keep at a very cold temperature until we need them. We'll then bring them out, we'll bring them into a tissue culture room, which is a room where we have a great deal of safety precautions, a great deal of safety equipment that allow us to work safely with the virus. We grow the virus on cells that grow in tissue culture. The virus, again, as I had said at the beginning, can't grow on its own. We need to have cells that it grows on. And so one of the things you'll see is somebody putting virus onto cells so that we can determine how well that virus is growing, either in the presence or absence of a small molecule, or to figure out what happens when we've removed a component of the cell to see if the virus needs that cellular component in order to work properly.
In our lab, when we grow viruses, we need to test them in cells in a specially contained environment. So we put them in different vessels like these plates so that we can see how well the virus can grow and infect and determine if we can block that infection using our drugs. We put our cells in incubators in special conditions where they can grow. We can come back the next day and see how the infection's proceeded.
This is one of our negative 80 degrees Celsius freezers. This is where we store protein extracts, viruses, and some of the different cells that we use in the lab. It keeps it at a very low temperature so that nothing oxidizes or degrades and is able to be stored for longer periods of time without it activating or doing anything weird. So it is a very cold freezer. So we have to specifically wear protective gear when we go inside and work very rapidly so that we can pull out the virus and keep it nice and cold for everything to remain cold. So individually chambered shelves, pull these out, and we're able to access individual viruses in their cryogenic tubes.
An autoclave is the way that we sterilize all of the waste that comes out of the lab. So anything that has viruses or cells in it, to make sure that it doesn't get out into the environment, is put in the autoclave. An autoclave is basically a very hot oven that gets very hot and also has very high pressure and steam. So it will kill everything inside the bag that we put in here. We also put in indicators to make sure that the autoclaves are actually working and that they kill and have the high enough pressure and steam, and there's nothing alive left in the bag. And so if I open it, you can see here that it's a very large area. We slide bags of our waste in. It goes for about an hour, and when it's done, everything is safe to go into the trash.
How do proteins change in the cell during infection? And in order to do this, we have to break open cells and then look at them individually, and that involves the gel that Jake is running.
And from that gel, once they're separated by their molecular weight, various other procedures can be done to analyze either the overall protein in the gel or to pick out specific proteins that have been contained within. And doing so lends insight into expression of cellular proteins or viral proteins that have been expressed within the cell, and aids in lending insight into how drugs are working or how well they are working.
You'll also see people counting plaque assays, which is a way that we quantify how well viruses grow. And essentially what you'll see them doing is they will have put a small amount of virus on to cells growing in tissue culture, and then they wait it a little bit. And then at the end of that time, they stain the live cells so that they turn purple. And in cases where a virus has gotten into a group of cells and killed them off, there will be a little hole in that purple staining, which we call a plaque. And that's an indication that there was a virus, it came in, it killed those cells, and that gives us an idea of how much virus is there. The more little holes there are in the purple stain, the more virus there was. The fewer, the less virus there was. And that gives us a way to really quantitate how much virus was growing. If we think we were killing that virus off, how well did we do? If you're a bacterium, you are able to live on your own, almost. Really what you need is food. If you're a virus, you need to take over something else. And it's that aspect of being an obligate parasite, requiring getting into somebody else's cells and taking that over, that is one of the things that interests my lab, is, how does this happen, right? What is it that the viruses have figured out that make them so successful at getting in and succeeding? But also in another aspect, we're particularly interested in understanding what happens when our cells win, right? Because it's a real competition. If the virus shows up and tries to get in, the cell's going to answer, I'd really rather you not be here. And so how is it that that battle gets fought, and how does it won? I think is one of the things we're really interested in. One of the things that we've probably all noticed is in the last 3-4 years, it seems that every 18 months, something like that, there's a new virus that is of concern. And so then the question is, where are they coming from? And I think my answer to that is best stated as, a lot of these viruses were probably here they were infecting some other animal. And we see them now as infecting people because these viruses have picked up a new trick. They've learned how to now infect a different animal, that animal being us. And as we have an increasing expansion of humans into places where they weren't previously in contact with animals that they weren't previously in contact with, this will probably continue to happen. But I also think even if we weren't expanding, this might still happen. Right? We spend a lot of time with different animals, right? We grow pigs, we grow chickens, and there are viruses that come from those animals that figure out a way to begin infecting us in a way that's harmful. And so I see that as something that will very much continue to happen, not only in the near future, but continually throughout the foreseeable future. How the virus penetrates the cell is usually it puts a protein on the outside of its assembled virion, or the actual virus particle, and that protein goes and attaches to your cell and triggers a set of events in that cell where now the cell brings the virus in. Once it has been brought in, the virus then uses that protein to essentially fuse to the inside of the cell and release all of its contents. So essentially it's discovered a way to bring a key that opens the door to the inside of the cell. When viruses have a key, they're looking for a very specific lock. Right? And so what that means in our cells is that the cells are making a protein that they're putting on the outside of our cells. And in HIV, for instance, the virus is looking for two separate proteins. And there are cases where there are people that lack one of those two proteins. In that circumstance, viruses cannot get into cells because part of the lock that they're looking for no longer exists. And so that's an example of how sort of this development of a virus to look for a specific lock, humans can develop mutations that make them now immune to these viruses. So how do people get viruses is a very difficult question to answer because it's not just one way, right? There are a lot of different ways that viruses end up traveling. And one is they'll come through insects, right? So mosquitoes are well recognized as things that transmit viral disease. Things like dengue, things like West Nile. Mosquitoes carry them, and essentially they are a very good machine for taking a virus and injecting it into our bloodstream. And so that's one way that this can happen. There are other viruses that have gotten very good at being transmitted just by us. So noroviruses, you'll know that there's regularly events on sailing ships and things like that where norovirus will spread very, very quickly because it has developed a way to be very easily transmittable between people. Flu is another example that's fairly easily transmittable. It appears to be able to be aerosolized very easily. But then there are other viruses that are actually really difficult to catch. Things like the new Middle Eastern coronavirus. That one does not seem to be very easily transmitted. Viruses like Ebola and Marburg, which have a very high fatality rate, do not seem to transmit really easily. You really need to have sort of direct fluid contact. And so it's not something where there's really a one-size-fits-all answer. It depends on what the virus is, how it gets transmitted. So what is global travel going to do to virus-based infections? And I think we're seeing it now, right? In the fact that the SARS coronavirus went from an index case in China to around the world fairly quickly, to the fact that now the Middle East respiratory coronavirus initiated in the Middle East, but now there have been cases that have been transferred in various different areas. And so I think that's going to be something that will continue to be a challenge, right? If this world is going to be connected more and more by planes and rapid transit, viruses go along with us. I think that is one of the reasons that continuing to do research understanding viruses and developing therapies and vaccines is important, because I don't think we're going to get rid of this event of new viruses coming, new viruses spreading. What happens when it is the Middle East and there is an annual Hajj and lots of people come from around the world to a place where it's known that the virus is hanging out? You're sort of tempting fate if you don't prepare for that to happen. So how fast do viruses mutate? They mutate really quickly. And if you were to compare to bacteria, I don't know that there's really a fair direct comparison, but influenza is an excellent example of viruses that are very good at mutating in an incredibly rapid manner. When antivirals are induced, there's a very rapid time frame of development of resistance. So I think this is going to be something that is very significant and is going to continue to be a problem. One of the reasons we're looking for how do you develop good small molecules is this is, I think, going to be a constant question of how do you do this? When one is developed, it will work, and it will probably be very successful at limiting disease and making people feel better, but it's not a permanent solution. It's going to be a step on the path of continually finding new tools, much in the same way that HIV has been sort of the most effectively treated with multiple different molecules at the same time. Right, so the two main workhorse viruses that I work on in the lab, one of them is called vesicular stomatitis virus. I often call it VSV, which is just the first letters of the three names that it has. And VSV has been used for at least 50 years as a research virus. It's not a human pathogen. Cattle and horses and sometimes pigs can contract it. But one of the reasons we work on it in the lab is, one, it's very safe for us to work with, because it's not something that I'm worried that my lab workers will get or that will be transmitted out into the community. But it looks a lot like a number of viruses that do cause disease. It's a cousin to rabies. There are a number of viruses that it looks very, very similar to. And so understanding how VSV works allows us to then extrapolate to these other viruses without directly having to work with them. And so that's one of the viruses we work with. Another virus that we've begun to do a lot of work with is called vaccinia virus. And vaccinia is the virus that was used as a vaccine. It was essentially the founder virus for the concept of vaccination, but it was used to eradicate smallpox from circulation on this planet. And it's currently the only example of a vaccine program that's completely obliterated a virus from circulation. And we've been studying this virus because it's similar to, it's similar to smallpox, which are eradicated, but it's similar to other pox viruses that are still circulating. One of which is monkeypox, which is endemic to Africa, and cases appear to be strongly increasing in the last few years as the smallpox vaccination has waned. And so we've been studying vaccinia to try to understand, what are the cardinal weaknesses of how this virus makes copies of itself so that we can apply that to things like monkeypox. And again, we're very pleased to use vaccinia because we see it as a safe laboratory virus that we can do a great deal of work with that can give us a lot of information about the true human pathogens without really endangering the workers and things like that. And so we've done a lot of work with those viruses and have sort of taken a couple different approaches. One of the things that my lab has had some success with is we've been trying to find small molecules that we can use to disrupt viral replication, right? How do the viruses get in and replicate is one of the things we're interested in, but the reason we're interested in that is we want to then come in and break those processes, right? Once we understand it, how do we define a tool that we can use that blocks that from happening? And so recently we found a couple small molecules, one of which hits the VSV type viruses that I talked about. And there's a small engine that this virus makes that makes copies of its genome. And so we're finding small molecules that block its function. And if it can't make copies of its genome, it can't make larger numbers of copies of itself. And we're doing that not only for that virus, but also for pox viruses, where we're looking at these engines and trying to find things to block it, trying to find wrenches to throw in the works so that it really doesn't work very well. One of the reasons that vaccines have worked so effectively for viruses, I think, is that vaccines are making use of your own immune system, right? They're making use of, your body's natural adaptive ability to come up with things that will now fight a virus. And so essentially, when you put together a vaccine and your body is exposed to it, your body looks at parts of an infectious virus and makes defenses against it. And it will often make defenses against multiple parts. Whereas, say, a small molecule really probably only targets a tiny little piece. Your immune system might be targeting 4 or 5 totally different areas within the virus and trying to block it that way. And it's much, much harder to evolve away from a 5-pronged attack than it is from a single-pronged attack. And I think vaccines often benefit from that type of approach. A similar approach we've been taking is slightly different, where we've been trying to address the question of, in the case of serious diseases, things with very high mortality. So these would be filoviruses like Ebola and Marburg, where mortalities can be 60%, whereas flu mortality is much, much lower, closer to 1 or 0.1%. In the cases where somebody has a very a virus that is associated with high mortality, one of the questions is, who else might that person have come in contact with that would also have this disease? And with those diseases, the thing that we know best is there are some treatments that look like they could be effective, but they're much more effective the earlier you give them. So we've also been looking at understanding how people's immune systems are responding to try to identify if we can track infection before you feel sick, right? To see if your immune system is giving you signals saying, yeah, I've been exposed to this disease in a way where you are not yet clinically sick, but you clearly have been exposed. That would allow us to really put a jump on starting treatment, right? So near neighbors of somebody who you know is infected, can you test them now and triage them for, okay, you should probably get some treatment. You look like you don't need it. And so that's one of the other things that we've been doing. That involves a great deal of computational analysis, a lot of looking at the entire expression patterns of immune cells to identify a few genes that are these canary in coal mine, these sentinels for identifying infection.
And we're trying to come up with methods to diagnose different diseases earlier than the way they're conventionally diagnosed using traditional methods. So to do that, we use bioinformatic techniques to analyze a bunch of data and come up with interesting patterns that tell us what type of virus a person is infected with. So for example, here I'm analyzing different types of viral infections with one is called Lassa virus, another one is called Marburg virus. And I've loaded data from multiple hosts that have been infected, and here I'm visualizing multiple genes and their level of expression, depending on what point those samples were collected. So here I'm looking at Marburg and Lassa samples infected 3 days, 5 days, 9 days. And by looking at this, I can compare the different types of expression of each gene and determine what the interesting patterns are and which ones are not that interesting. So this is an example. This is the Ebola virus. And this is something we call a microarray. It has 45,000 probes, and each probe has a fluorescent tag. And depending on the color of that probe, we measure the amount of expression. So all of these little dots represent an individual probe, and the color indicate the amount of expression that that probe has, which correlates with the amount of expression that a specific gene has. So genes that look red have a higher expression than genes that look green. And then we can generate these types of plots with similar information. So here I'm showing 24 samples from multiple conditions depending on the color. And those are the sample sizes that we've been working with. But each individual has 40,000 or more data points.
We are looking at new ways to detect viruses. And the idea is we want to make diagnostics as simple as possible. And currently, a lot of the technologies that do the best diagnosis require a lot of preparation, require you to take a sample and make nucleotides from it, and then you need to amplify those nucleotides, and by doing that you can identify the infectious agent. You can identify what's making you sick. We are looking to make a silicon-based chip with a simple sort of LED-based detector, where now we can just flow blood or serum over that chip, and if there's virus present, it will get captured, it will get visualized, and we can say, aha, we can see that what you have looks like influenza, and that can speed treatment. And the simpler it is, the more likely it is to become point of care, something that can be very simple to use, very simple to understand the results to. And so that's something that we are currently working on. And in the laboratory, you will see one of the prototypes that we have constructed, where essentially what we've reduced it to is a fairly small instrument with a very tiny chip, where we can just place the chip on the reader, hit go, and the reader takes care of analyzing that diagnostic chip and telling us what viruses are present, what viruses are not present. And one of the things that's really an advantage of this technology is we can ask several questions at once. Right? So we can say, we're now looking over, say, 10 viruses, 12 viruses. Do you have any of these? And might you actually have a couple of these? Because that would change how you would treat someone who's sick. If you can determine that it's one or two, and not be stuck in a paradigm of just trying to find one, but to try and understand the totality of the infection and of the disease.
This chip would have antibodies on it that are specific for a virus. If you want to know if the virus is in the sample, you place the chip in the sample, and then if the virus is there, it should bind. You load the chip onto the platform, and then click Scan Chip. And it will draw the chip into the machine and detect if there is signal for virus.
And what you're also seeing in that image is that there are pieces of junk on that spot. What you won't be able to see very clearly is there are much smaller points of light that are the viruses themselves. And the technology has been set up to differentiate between junk and virus, which is actually that's one of the key aspects of being able to separate those two. A virus encounters a cell and tries to get in, and the cell actually recognizes it and shuts it down. How does it do that? If you look at a cell using a microscope, you can see it developing a certain type of response. So what will happen is it will move proteins around in the cell, proteins that had been sort of diffuse, they weren't really anywhere in particular, will all of a sudden begin forming physical cages around sites of virus replication and shutting down virus replication. And so we've been very involved in using microscopy and looking for various different proteins, which involves using various different labels at various different colors in order to look for where those things go and where do they go relative to where the virus is. And identifying what those proteins are and what they're doing, we're looking for ways to stimulate that so that outside of the lab, we can find ways to make that natural defense much more enabled, much more capable. And in a sense, that would be a really nice way to fight viral infection if you can just sort of wake up a latent defense in your body and say, now it's time to turn on because we're pretty sure there's a virus around.
So this is a fluorescent microscope. We use this to image cells that have been infected with viruses, and we're able to do that by not only using live virus to see how it enters the cells and propagates throughout the individual cells and spreads throughout cells. So we mount the cover slips that these are grown on onto glass slides, and they're mounted directly into the microscope. Okay, so we've mounted the slide onto the stage here, and it's hit with fluorescent light that's then absorbed by these specific labels that we have stained the slide with, and that is going to let us look at the cellular nucleus. It's going to let us see where the viruses are, and then it's going to also look for some specific proteins that we're interested in studying how they relocalize within the cell. So we typically capture a sequence like this of all three of the channels and then layer them together later. Composite, you can see that it's been colored so that the nuclei are blue, the cells are green, and then the infected cells are the red ones. And we can actually look at the shape and the localization of those viruses to compare what our different treatments are doing.